Good afternoon, my dear students and friends. Myself, Dr. Ravikant Pathak, and I'm going to start your video lecture class. In the last video lecture, I have discussed about the glomerular filtration and glomerular filtration rate and different pressure which involves in the favors filtration or oppose filtration. And today I will talk same topic, but different. Segment. So you can say different segment means second portion of this topic. So here we are going to discuss, because I had already told. So there is the two different process. One is the chemical process, another is the mechanical process. Chemical process urea cycle, mechanical process that is the formation of urine, in which you have to see there is the three different methods. So first is the glomerular filtration, second is the selective reabsorption, and third is the selective secretion or tubular secretion. So in the last video lecture, I have discussed about the glomerular filtration and glomerular filtration rate. And in this video lecture, I will talk about the second portion of this topic, and the name of this portion is, you can say, that is the selective reabsorption. So we will see where is the selective reabsorption takes place in the different parts of the nephron. And one more small topic, you can say, that is the JGA, Juxta Glomerulus Apparatus. So now I am going to start. So here you can see. First is that they have given about the Juxta Glomerulus Apparatus. So JGA. However, I have already discussed a little bit about the glomerulus region in the last video lecture. But eventually, I am going to here start about the JGA. JGA, before I am going to discuss, you can see in the figure. So this is the JGA, Juxta Glomerulus cells. So here they are showing the Juxta Glomerulus cells. If you will see the position of Juxta Glomerulus apparatus or Juxta Glomerulus cells, so this is the afferent arteriole, and this is the D.C.T, distal convoluted tubule. In between it is present. In between it is present. So they may be asked in your exam. So where is the Juxtaglomerulus apparatus located? So Juxtaglomerulus apparatus, it located in between the D.C.T as well as afferent arteriole. So this is the actual location. Another thing, what is the role of Juxtaglomerulus apparatus? So Juxtaglomerulus apparatus, when the glomerulus filtration rate become slow or fast, so that time they secretes the substance renin, and by which they can maintain the glomerulus filtration rate. GFR can be maintained by this GGA apparatus. So this is the basic function, and we will see and we will also read in this slide. So here you can see. First, they have given about the juxtaglomerulus apparatus. It is saying the juxtaglomerulus apparatus is the part of kidney nephrons. It means this is the part of kidney, and kidney basically nephron you know very well. This is the structural and functional unit of the kidney. And followed means after that, followed to the glomerulus region. If you will see, this is the glomerulus region. After the glomerulus region, so there is the juxtaglomerulus apparatus located. And where it located? It located means it is found between afferent arteriole and distal convoluted tubules on the same nephron. It means single nephron. In the single nephron, so in the single nephron, this is located. Means you can say this is single nephron, and in the single nephron between afferent arteriole, afferent means which brings the blood inside the glomerulus region. So afferent arteriole and distal convoluted tubules, in between these are the present. Okay, and what is the this location is relevant means they are related. So this location is related to its function in the regulation of the renal blood flow and the glomerulus filtration rate. It means they regulates the GFR as well as the renal blood flow. How regulate? So here you can say they have given about the mechanism, how it acts. So a fall in the glomerulus filtration rate. Suppose that if the glomerulus filtration rate, we have to already studied in the last video lecture. So 125 ml per minute, and 180 liter per day. So this is the GFR, actual value of the GFR, glomerulus filtration rate. So here it is saying when the glomerulus filtration rate fall, a fall in the GFR that activates the cells of the Juxtaglomerulus apparatus. And these cells, they have started to secrete. They have started to secrete renin. Renin is the hormone. Don't get confused, because renin we have already studied in the digestive chapter, digestive system chapter. So there is also stomach secretes the renin, but renin generally found in infants, not in the adult or a young means mature person. So renin generally secretes infants, but these renin because they have the similarities, they have the little bit similarity. There is the double NIN, and here only the NIN. So renin, so these renin secreted by the juxtaglomerulus apparatus, and these juxtaglomerulus apparatus when secretes the renin, by which a through of series reaction occurs and brings the GFR back to the normal. Means when this if the glomerulus filtration rate become fall, so that time JGA secretes the renin, and renins will brings the normal means you can say they brings the GFR to in normal position. It back in the or brings in the normal position. This is the function of the JGA. So this is the story about the JGA. Now we will see second topic, second portion of this topic, and second portion, that is the tubular reabsorption or selective reabsorption. So here we will see reabsorption. Okay, reabsorption means you know very well. So that is nephron is the, they have the different length and the different parts, they are already divided, like P.C.T, loop of Henle, D.C.T, collecting discs. So in these parts, they are liable for the absorption of the different substance and secretion of the different substance. So this is the second portion of this topic, and third portion I will discuss in the next video lecture about the tubular secretion. So here I will only discuss about the reabsorption of the tubular reabsorption or selectively reabsorption. So here what is the story about so reabsorption firstly discovered by the scientists, and those scientists who discovered, so here you can see who discovered the reabsorption. So reabsorption was discovered by the Richard and supported. So scientist name is the Richard. They have discovered the reabsorption phenomenon. So reabsorption phenomenon was discovered by the scientist Richard and their supporters. How many percent? So it is saying nearly 99% of the filtrate is reabsorbed by the epithelial cell lining of the renal tubules, because we have to see in the glomerulus filtration rate. So glomerulus filtration rate, everyone will forms only 0.8%. 0.8% forms the urine. So that means you can say 0.8% means less than 1%. So that's why it is telling about that here. Nearly 99% of the filtrate, 99% of the filtrate reabsorbed by the epithelial cell lining of the renal tubule, and some substance they can absorb through the actively. So actively like amino acid, because charge molecule, every charge molecules we have to should create a basic concept. Every charge molecules they can absorb by the actively, but some can also absorb the passively, like chloride ions and glucose molecules. They can also absorb the passively. So here glucose they have given actively, so sometimes glucose can be absorbed the passively. Here why we are saying actively, because during the secondary active transport, glucose and sodium both will go together. So that's why it is discussing about the actively. And this is the story about the reabsorption. So now we will see detail first reabsorption in the P.C.T, means selectively reabsorption in the P.C.T. So here they have given the selective reabsorption in the proximal convoluted tubules. So this is the very important, and they already asked in your NEET exam. So the pivotal means basic, fundamental, or you can say simply the basic site of the reabsorption is the P.C.T. Means maximum reabsorption takes place in the proximal convoluted tubules. Nearly all of the essential nutrients and 70 to 80% of the electrolytes and water are reabsorbed by these proximal convoluted tubules. Now here you can see, means most of the reabsorption generally takes place in the proximal convoluted tubules. Active uptake of ion reduces the concentration of the filtrate and equivalent amount of water passes into the peritubular capillaries by osmosis. It is saying, suppose that how you'll create the concept. I'm drawing the figure. With the help of figure you can understand. So this is suppose that this is your glomerulus region. This is your glomerulus region, and this is the uh, different loop of LA, and then further here ascending and descending, and furthermore there is the D.C.T, and ultimately it forms the uh, collecting duct CT. So D.C.T and CT. So here you can see. Now in this figure, if you see, so these have drawn, and it is saying, so active uptake of ions reduces the concentration of, because you know, so in this P.C.T region, so there is the active, so molecules reabsorbed actively, reabsorb. So, I'm drawing the figure. So this is the peritubular capillary. This is, you can say, peritubular capillary. And here saying, so these ions enters inside the peritubular capillaries by actively, and you know, when the salt will move, that time water will also follow them. Water will also follow them, because salt is going front, salt is going ahead. After that, water also following them. So such type of the movement is known as the obligatory water reabsorption. So such phenomenon also known as the obligatory water reabsorption. Why we are saying obligatory water reabsorption? Because when the salt will move inside the peritubular capillaries, water molecules also will follow them. So that's why they have given the obligatory water reabsorption. So I think you are understanding. So suppose that this is the molecules, and here this is the salt molecules. When salt molecules enter, so when salt molecules enter inside the peritubular capillaries, after that water molecules, so water molecules I'm representing like this. So water molecules also enter into the peritubular capillaries. So such type of the phenomenon is known as the obligatory water reabsorption. So here they have given. So here 80% of water is reabsorbed by the passive transport. Why? Because most of, here it is saying, ions reduces the concentration of filtrate and equivalent amount of water passes into the peritubular capillaries by osmosis. Because when the actively means with the need of energy, water molecule with the need of energy, electrolytes enters into the peritubular capillaries, and water molecules also following that path, and due to this phenomenon is known as the obligatory water reabsorption. Most of the important 80 to 90% buffer bicarbonate is also reabsorbed from the filtrate, because we are discussing here selective reabsorption. So, and these bicarbonate, why they have given? Because it involves play important role to maintain the pH. So that's why 80 to 90%, approx 80 to 90% buffer bicarbonate is reabsorbed in this region. So means in the P.C.T region they also absorb. Some urea is reabsorbed by the diffusion. We know very well. So urea movement generally takes place via the diffusion, because this is the organic molecule, so they can easily diffuse through the cell membrane or plasma membrane. The rest remain in the filtrate are removed in the form of urine. So here it is saying in the urine, not actually, because glomerulus filtration forms after that when enters into the Bowman capsule, that is called nephric filtrate or glomerulus filtration. So when it after that it will pass inside the P.C.T, and at P.C.T region you can't say urine. Urine you will use, urine term you will use only the D.C.T, distal convoluted tubules. And when you will use, after the tubular secretion, when the tubular secretion completed in the distal convoluted tubules, so that time you can use the term urine. Okay, before you can't say urine, okay. So they have given some urea reabsorbed by the diffusion, the rest remain in the filtrate form, removed in the urine. Okay, they removed in the urine, but you can't say that is actual urine, because it will pass through the loop of Henle, then D.C.T, and D.C.T tubular secretion completed. That time you can give the term you can give the term urine. So actual that time you can say this is the urine. Now we will see selective reabsorption in the loop of Henle or simply Henle's loop. I'm going to draw the figure. So with the help of figure, you can understand. Suppose that this is the loop of Henle, and we know very well. So there is the two different types of the loop of Henle. One is the descending, and another is the ascending. In the descending, before I'm going to discuss this topic, this statement or simply this slide, you should to make this concept. What is the concept? This descending loop of Henle, this will be permeable to water. This will be permeable to water, but impermeable to electrolytes. This should create the basic concept, because I will discuss in the video lecture. I will also further discuss counter current mechanism. So these loop of Henle play very important role during the counter current mechanism. So right now you should make basic concept about the loop of Henle. So here this loop of Henle generally U-shaped structure, and descending loop of Henle, it is the permeable for water but impermeable to electrolytes, and just inverse of this into the ascending loop of Henle. It is the impermeable to water but permeable to electrolytes. It is impermeable to water and impermeable to electrolytes. If you'll see, so in the P.C.T region, so there is the osmolarity 300 millius, because this is only found in the plasma. So these plasma, they have the 300 millimole osmolarity. And if you'll move from upward to downward, means in the descending loop of Henle, so what will happen? Their osmolarity will increase. Why osmolarity will increase? Because water is going to outside. Water is passively going to outside. That's why when we'll move from upwards to downwards in the descending loop of Henle, so the millimole osmolarity will decrease, and solution will be, or urine, simply you can say, you can't say, I had already told you can't use the perfect term urine. So you can say filtrate. The filtrate is become highly concentrated, and now when the filtrate is moved into the descending ascending loop from downwards to upwards, it will become the hypoosmolar. It will be hypoosmolar. Why? Because salt removes the actively outside, and water will not enter. So that's why their osmolarity will decrease. That means you can say filtrate become hypoosmotic. Okay, this is the basic concept you should to create. And now I'm going to discuss this slide. Now in the slide you can read, they have given the reabsorption in this segment is the minimum. Here we have to see in case of the P.C.T, maximum reabsorption. P.C.T proximal convoluted tubules, there is the maximum reabsorption, but in this region, means loop of Henle, there is a very least, means least reabsorption takes place. This region plays a significant role in the maintenance of the high osmolarity of medullary interstitial fluid. You should to know there is the very important role of the loop of Henle to maintain the osmolarity of the filtrate. The descending limb loop of Henle is permeable to water but almost impermeable to electrolytes. That's why I had already told. So water can be moved into the descending loop from descending limb loop of Henle to interstitial fluid or peritubular capillaries. But and this concentrates the filtrates and it moves down. And I had already discussed, when we'll move in descending limb of loop of Henle from upwards to downwards, so that time this value means their osmolarity will increase and filtrate will hyperosmotic. Okay, and just inverse happening in the case of the ascending loop of Henle. So now here you can say the ascending limb is impermeable to water but allows the transport of electrolytes actively or passively. So they can be moved by both actively or passively, but you should to know this is impermeable to water, this is impermeable to water. And since as the concentrated filtrate pass upward, and it gets diluted due to the passage of electrolytes to the interstitial fluid, or simply you can say medullary fluid. So when the electrolytes actively or passively pass inside the interstitial fluid, so that time their osmolarity will decrease, and when osmolarity will decrease, the solution or filtrate become hypoosmotic in nature. So this is the story about the selective reabsorption in loop of Henle. So here you have to see there is the minimum reabsorption, but what is the basic function? The basic function to maintain the osmolarity. So this is the basic function of loop of Henle. Now here in this figure you can see different regions of the nephron, and where these substances is secreting, which substances is absorbing, reabsorption takes place. So here you can say in the figure. So with the help of figure, I'm going to discuss. Firstly, this is the GFR, glomerulus filtration rate. We have already studied in the last slide. And here you can say, if you'll see upward, these are the reabsorption. These substances get reabsorbed. These substances get reabsorbed. And downwards, this is the secretion. This is the secretion. They are saying about the secretion. Secretion of hydrogen, secretion of ammonia, secretion of potassium, and secretion of the hydrogen. Why these are secreting? Because hydrogen ion maintains the pH of the filtrate or urine, and potassium and sodium, potassium ion maintain the sodium balance, means ions balance of the urine. So now this have given. So this is the if you'll see in the figure. So first they have given the glomerulus region. This is the P.C.T, where is the maximum 80 to 90% reabsorption takes place. This is the loop of Henle U-shaped. There is the no, means at least, means you can say least, not at least, least absorption, least reabsorption takes place. But here you will see, so this is the descending loop of Henle. Descending loop of Henle, where is the waters get outside, because this is the permeable to water, but electrolytes can't get outside. Due to this, their osmolarity will increase. Suppose that here 300 milliosmolarity, so it will goes to downwards, then it will become the 1200 millimole osmolarity. In this way you can say, when we'll move from upward to downwards into the descending limb loop of Henle, so the osmolarity of the filtrate generally increase, and this solution you can say hyperosmolar. And after that, in case of the ascending limb loop of Henle, so there is the just inverse takes place, and osmolarity of the solution generally decreases. This is the basic concept. And after that, here they have given the D.C.T, distal convoluted tubules, where the secretion of these ions takes place, tubular secretion, and you can use the term urine here. And this is the collecting ducts. In the collecting ducts, everything absorbs, and some urea also pass out into the interstitial fluid, and water for the reabsorb. Means you can say in the collecting duct only those urine substance, those substance will loss, which is not necessary to body, and there is the very less loss of the water takes place. So this is the story. This is the total story about the selective reabsorption as well as selective secretion or tubular secretion. We will discuss in the next video lecture about the tubular secretion. So now here you can see in this selective reabsorption, the D.C.T, distal convoluted, how happen in D.C.T means this is the D.C.T region, distal convoluted tubules. So conditional reabsorption of the sodium and water takes place in this segment. So here you can say NaCl, that is the sodium, and here water and bicarbonate also absorbs. And in compare, what is generally secrete? So D.C.T distal convoluted also capable reabsorption of the bicarbonate ion. So here in the figure you will see reabsorption of the bicarbonate ion. And selective secretion, that means you can say tubular secretion, selective secretion or tubular secretion of the hydrogen ion. So this is the hydrogen ion, and potassium ion, here potassium ion, and ammonia to maintain the pH and sodium potassium balance in the blood. So due to this, in the D.C.T region, distal convoluted region, so there is the three substances secrete: potassium ion, hydrogen, and ammonia. And these hydrogen ion to maintain the blood pH, while ammonia and while potassium, they maintain the sodium potassium content or concentration inside the urine or inside the blood. So this is the story about the selective reabsorption in the D.C.T. Okay, so this I have given about the selective reabsorption into the D.C.T. And now I will talk about the next, that is the selective reabsorption in the collecting ducts, where the this is the last part of the nephron where the you can say finally urine formed. Here urine formed. So this is the long duct extend from the cortex. This you will see this collecting ducts. This is the very long, not very much long but long. So this long duct extend from the cortex of the kidney to the inner parts of the medulla. These collecting ducts, they have extended from the cortex. You have to see cortex is the outer part of the kidney and medulla is the inner part of the kidney. So these collecting ducts will extend from to the cortex part of the kidney and in up to the region of medulla. So inner parts of the kidney. So here they will so extend cortex of the kidney and inner parts of the medulla. Large amounts of water could be reabsorbed from this region. I had already told, in the collecting duct there is the fully urine formed and there is no water will loss. There is no water will loss. So they have given large amounts of water could be reabsorbed from this region, produce a concentrated urine. So it means in the collecting ducts there is the highly concentrated urine generally pass outside the body. So now, and this segment allows passes of small amounts of urea into the medullary interstitial to keep up the osmolarity. So means in the collecting ducts. So because they are passing the urea, we have to see maximum reabsorption takes place in the proximal convoluted tubule, but some certain certain urea remain inside the filtrate. After then when it will arrive into the collecting ducts, so here in case of the collecting ducts, so urea further reabsorbs. So urea further reabsorbs. Where it will go? It will go into the medullary interstitial fluid, and when the urea comes inside the medullary interstitial fluid, to maintain the osmolarity of the filtrate or maintain the osmolarity of urine. So you can use here term urine because this is the collecting duct. So here to maintain the osmolarity of urine. It also plays a role in the maintenance of the pH and ionic balance of the blood by the selective secretion of the hydrogen and potassium ions. So we have to see every time secretion takes place, three component: ammonia, potassium ion and hydrogen ion. So here in case of the collective duct, also secretion of the potassium ion, hydrogen ions, which will maintain the pH as well as ionic balance of the blood. So now this is the we have to discuss today about the second portion of this topic, and we are studying about the mechanical process of urine formation. Means, so simply you can say mechanical process or urine formation. So we have to study two things. First is the glomerular filtration, second is the selective reabsorption, and in the next video lecture I will discuss about the tubular secretion and these three different way by which complete urine formation takes place. Okay, so thank you, and everyone should share, subscribe my channel, and hit the bell icon for regular updates. Okay, so now here you can say thank you for watching, and everyone should share, subscribe my channel. Okay, so thank you again.